Welcome, everyone. In today's lecture, we're going to be looking at 4 different types of tissues. When we look at the 4 different types of tissues, you'll see that we have epithelium, connective tissue, nervous tissue, and muscle tissue. Now, I believe in a previous lecture, I covered muscle tissue because we spoke about smooth muscle, skeletal muscle, and cardiac muscle. I will show you neural tissue. That's actually not very much to cover. But epithelium and connective, these two are a little bit more challenging. There's a lot of subcategories under each of them. One of the big differences between epithelium and connective, if I were to draw a bunch of cells here that we're looking at under a microscope, and these look like cubes, notice they're very closely packed together, right? And if I make it a few layers, again, these cells are very, very closely packed on top of one another and close together. There's very little extracellular matrix in epithelial tissue. Lots of cells closely packed together with very little extracellular matrix. Connective tissue, on the other hand, maybe we have a red blood cell here. Maybe we have a lymphocyte here. Maybe we have a monocyte here, right? We have cells, but they're not closely packed together. And we have lots of extracellular matrix, right? We have lots of liquid and plasma. In this case, if we're talking about blood, it's the plasma, which is the extracellular matrix. And in connective tissue, there's lots of it, lots of ECM, and the cells are not closely packed together. Those are 2 very important features. Tissues all have distinct structure. And if their structure or form is different, then their function is going to be different, right? Basic principle of anatomy and physiology: structure determines function. And tissues are important because tissues come together to form organs. Just like chemicals come together to make cells, cells come together to make tissues, tissues come together to make organs, organs come together to make systems, and then the 11 systems make the organism. When we're studying tissues, we call that histology. When we're studying cells, we call it cytology. When we look at epithelium, epithelium is the type of tissue that's going to line things. It's going to cover things, especially the external surface of the body. When we talk about the external surface of the body, what comes to my mind is the skin. And the layers of the skin that you see with the naked eye is the layer on the above, which is the epidermis. Remember, epi means above. There's epidermis. And then, of course, we just have dermis, which is deep to it. So the epidermis is a type of epithelium. It's covering the external surface of the body. And then we also have epithelium that lines body cavities and tubules within the body. A tubule is, as it sounds, anything that has or looks like a tube or a tract, whether this is the trachea, whether this is the esophagus, whether this is the small intestine, large intestine, arteries, veins. They're basically just tubules or tubes. So we'll find epithelium that lines tubules and even body cavities, as well as glands. Epithelium is designed to protect us. If we think of the skin, it protects us against excessive friction or bacterial invasion, and even chemical damage. Certain epithelium have cilia on it, and really cilia are just these hair-like extensions like this that we'll find at the end of, let's say, an epithelial cell like this. There's the nucleus. So we have cilia on the top, and these cilia can beat rhythmically, and they have this sweeping action. So let's say, see if I can draw this here. let's say here is parts of the trachea, the windpipe, and we have these cells along the side here. They look like tall columns, so we're going to call them columnar cells. And we have some on this side too. I just don't have enough room, so I'm just going to draw them on one side. And then at the very end, we're going to have these hair-like cilia this way here. These are going to be the cilia. And those cilia are going to be beating upwards rhythmically from inferior to superior. Remember, you're breathing in air, and air and dust particles are coming in this way, but the cilia are going to beat upwards. And then we also have, let's say, these other cells. I'm going to make them blue here. These are called goblet cells. And goblet cells are just hidden in here. But what they do is they secrete mucus. And that mucus is going to go to the top where the cilia are, and it's going to create this layer that's going to stick to the cilia. So that when you're breathing in air that has dust and debris and foreign particles, rather than have all that debris make it bypass all of that and make it into your lungs, it's going to stick to the mucus. And then all you have to do is like that, and you can cough and get it out of your system. So the cilia right here, these are really important. They are found in the respiratory tract, and they sweep any dust or foreign particles away from the lungs. You'll also have these cilia in the woman's fallopian tubes or the uterine tubes that are moving and trying to move the ovulated egg to be fertilized by the sperm. Again, the sweeping motion of the cilia. Epithelium is also used for absorption. We have absorptive cells that we find in the stomach and in the small intestine. And we have smaller cilia there. Again, hair-like structures, but they're smaller than cilia, and we call them microvilli. And the purpose of these microvilli is that nutrients are going to be absorbed by them. They're going to act like a sponge, and sponges absorb and bring in things. So epithelium is good for protection. It's good for absorption. We find them throughout the kidneys, and the kidneys are really important to filter, excrete, and secrete. So the kidney tubules, when you get into anatomy and physiology too, and you learn about something called the nephron is the functional unit of the kidney. The nephron has a proximal convoluted tubule. And if there's a proximal convoluted tubule, it's going to have a distal convoluted tubule. And the proximal and the distal convoluted tubules, again, there's that word tubule, remember? Epithelium is found in tubes or tubules. We're going to find epithelium in the proximal and distal convoluted tubule, especially cuboidal cells. They look like cubes. Those are cuboidal. These are columnar. They look like columns. Okay. They're used for filtration, excretion, and secretion, right? What are they secreting? Glands secrete substances, just like this goblet cell secreted something, okay? So cilia we covered, and microvilli we covered. Cilia are these longer, larger extensions that come off the cell or even the plasma membrane, and they help to move secretions and fluids across the surface, just like the goblet cell secreted mucus and it was sitting along the cilia. Microvilli are much smaller projections. That's the sponge that's used to increase absorption. So we're going to find them along the intestines. Microvilli are really important. People that have celiac disease or sensitive to gluten. Again, if this is part of the small intestine and we have these columnar cells that have these very small microvilli, the importance of these microvilli is they're the sponge that's going to allow the nutrients to be absorbed And then over here, we're going to have our blood vessels. You're going to have these small little blood vessels, and the nutrients that are absorbed by the microvilli are going to pull them into your circulatory system to bring the nutrients to all the parts of your body for energy. So people that have issues with the microvilli, maybe they have celiacs, maybe they have gluten issues, the microvilli here get broken down. So these people do not get to absorb all their nutrients properly, and creates malabsorption or malabsorption diseases. So when we look at epithelium, we have to ask ourselves three questions, okay? So 1, 2, 3 important questions when labeling epithelium. Number one, when we look at it under a microscope, we have to say, do these cells look like they are closely packed together? Do these cells look like they're closely packed together? If they are closely packed together, then it's a type of epithelium. But if they're spread far apart, we can't call it epithelium. So question number one is, are the cells close together? And if they are, the next question is, okay, are the cells one layer thick or more than one layer thick? If it's one layer, we refer to it as simple epithelium. And if it's more than one layer, we call it stratified epithelium. So if we look here on the top, this is simple squamous, this is simple cuboidal, and this is simple columnar. They're all one cell layer thick. If it's more than one cell layer, then we call them stratified. More than one cell layer, stratified. The third question we ask ourselves is, hey, what's the cell shape? If it's this flat, irregular shaped cell, it's called a squamous cell. If it looks like a square or a cube, then we call it a cuboidal cell. And if it looks like a tall building or a column with a nucleus, then we call it columnar. Okay, so are the cells closely packed together? If it is, it's epithelium. If it's one cell layer, we use the word simple. If it's more than one cell layer, we call it stratified. Then we have to look at the shape of the cell: flat and irregular, squamous, square-like, cuboidal, and column-shaped columnar. So if we go here, again, we see these flat, irregular shaped cells with these nuclei, and it's one cell layer thick, so it's simple squamous epithelium. Because it's so thin, it allows things to pass through easily, like oxygen, or things passing the other way, like carbon dioxide. So we can find these in the lungs. It allows for certain things to diffuse. We call it diffusion, when it can easily pass through this filter that's very, very thin, simple squamous. But below it, we see it's stratified squamous, right? Same flat, irregular cells, except we have multiple layers towards the apical surface, or the top surface. So where we have the epidermis of the skin, right? Your skin, epidermis, we have many layers, because if you scratch yourself with your nails, could you imagine if it was only one cell layer thick, you'd have that one layer under your fingernail, and then you'd start bleeding. So we want many, many layers of protection on your skin. So stratified squamous epithelium. Over here, we have these cube-shaped. You can see they're squares. They're closely packed together. So it's epithelium, simple cuboidal. And then we have many layers of that called stratified cuboidal. The simple cuboidal is found in the kidneys, especially at the proximal convoluted tubule and the distal convoluted tubule. Okay. The stratified cuboidal is found in other glands, like certain sweat glands will have stratified cuboidal. Then to the right of that, we see one cell layer thick of tall columns, so it's simple columnar. And then look at the one down below. It's kind of tricky, right? Because it looks like columnar cells, but if you look above, you see how these nuclei are all at one level. So it gives it that appearance that it's one cell layer thick. But if we look down here, looks like these are down here. These nuclei are at this level. Then we have some at this level. So because the nuclei are at multiple levels, it gives it the appearance that it's stratified, but it's not. It still is one cell layer thick. So we call it pseudo-stratified. And we'll see pseudo-stratified lining the trachea, parts of the respiratory tract. And that was what I drew before, where we had these columnar cells like this with the cilia coming off of them. So sometimes they call it pseudo-stratified ciliated columnar. And if someone's a smoker and they're inhaling smoke, then what happens to these cilia is they degenerate and they break down. And smoke and all the carcinogens and chemicals within smoke that paralyze the cilia or that break down the cilia, now you don't have anything trapping the debris. So all the chemicals make it down into the deeper part of the lungs. And when the cells and the tissue within the trachea or the respiratory tract break down, when those pseudo-stratified ciliated columnar cells degenerate and break down, your body replaces them with something else. And whenever the body takes one cell or tissue and breaks it down and becomes something else, it's never, ever a good thing. It's actually morphing. It's changing its shape. There's like a metamorphosis that's taking place. And then they become squamous cells. They become squamous cell, and you've heard of squamous cell carcinoma, which is a type of cancer. Okay. Then we have transitional cells. These transitional cells, they alter in shape. Think of a deflated balloon, right? If you were to put the letter, let's say you draw a square. Let's say you draw a square on a balloon that's, let's say, deflated, and you put a square like right there, but then you blow it up and you inhale. Isn't it safe to assume that the shape of that square starts to modify and maybe becomes a circle or it becomes flat looking? It just changes its shape. So if this is the urinary bladder or this is any part of the urinary tract, it can be inflated, right? The urinary bladder can be filled, or there could be nothing in it at all, and then it changes its shape. So the cells can alter in their shape. We call them transitional cells. We find that anywhere in the urinary tract. Okay, so again, we have simple epithelium, stratified epithelium. We have squamous, which are irregular shaped. Those are good for protection, secretion, and even diffusion. We spoke about oxygen and CO2, carbon dioxide. Then we have cuboidal cells. We said we find them along the tubules of the kidneys, the proximal convoluted tubules and the distal convoluted tubules. And then we have columnar cells, which we can find in the respiratory tract and even the digestive tract. Again, when we look at the cells, we're looking at the cells that are close to the apical surface, which is the top. Or if we're looking at a cross section of a lumen, let's say it's a blood vessel, we're looking at the cells that we can find right there, right? Like that. If we take this cross section and we cut this out, now we're looking at something that may look like this. And now we're looking at the cell layer at the apex at the top, which would be right there. Those are the ones that we'd be studying in terms of their shape and how many layers. Pseudo-stratified, remember, it's not really stratified. It's a simple columnar, but because the nuclei are at different levels, it gives this false appearance that it's stratified. And wherever we have pseudo-stratified, we'll often find that there are cilia located there, especially in the respiratory tract or the trachea. The transitional is a type of stratified squamous, but we find them in the urinary tract, in the urinary system, whether it's the bladder, the ureters, or even the urethra, where the cells could be flattened when the organ is full, or they could be rounded when it's empty. So think of that balloon that alters and modifies in shape. So under simple epithelium, we have these three, and under stratified epithelium, we have those. Okay. Okay, we'll take a quick break here. We'll come back. We will look at connective tissue.